So good. People use this mostly. Let's try this out. When people come in, we'll get their presentation on this and we'll load it on here.
Yep.
Let's see. So
Yeah. Okay. Okay.
if you see a little metal clip, it fell off my. I have my phone in my. So I have this like. Right. Yeah, I mean, you could get new metal. I got another. The dean's address in case. Just when the speakers come in, hopefully they'll come in soon. Tell them to come right over.
Yeah.
I might shift up more. Just gonna be late. Hello, good. All right.
Oh, maybe he was doing something. Okay, whatever. I'm sorry, what's your name?
Benedetta.
Benedict.
Sorry.
It's
Okay, that's okay.
it's the one that says BD.
This one?
Yes. Is there time to try and opening it as
Yeah,
well?
let's do that.
And
You want me to actually display it or?
can we try the presenter mode if possible?
Are you a student? Postdoc?
Student, PhD student. Yeah.
PhD student.
Yeah. I think like slideshow
Yeah, you should.
and
You want it so you can see your notes?
that would be the best if possible.
Do you
But
need
I
it?
don't know if for people. No, I mean
You
I've
shouldn't
rehearsed.
need it.
Okay, yeah, no, I mean I've rehearsed without, but
I'm sure you're gonna
Yeah.
be fine. But when we go
Okay.
to, let's see. Let's see. I'm trying to think of how it's automatically going to display it.
okay.
It's not going to do it that way. Let's see.
That's-
Custom shows, setup slideshow. I just want to screw up your thing. How do you normally do? I've never used presenter notes in my life. I have no idea
Normally,
how you even do
okay,
it.
yeah. I mean, normally,
Can I search right here?
normally it has like
Always under this icon.
Yeah.
Like a computer setting where you like do a shared display.
Like,
It's
when-
mirror display.
It's currently not set up so they can see presenter notes, but I think there's a call for wanting to see presenter notes. So I'm trying to make it so that when it presents, it's automatically bringing up presenter notes. But that's the face I made. I'm like, I
I mean,
don't
if
know.
it doesn't, it doesn't. It's just, I think, in general. Maybe they're just not enabled on.
No, I'm sure it is. I just don't know how to do it.
Because normally what happens is,
But it says use presenter view.
It's not doing it because there's the live stream running.
So we can't do both.
Can't do both.
Okay.
We have the live stream running.
that's
Okay.
okay.
Do you want to see it? Is that good? Are you
Yeah,
set?
yeah,
Okay.
yeah.
Okay. And
So
then,
minimize that. This is Marilena,
Benedetta.
Teresa, and Penelope. Okay, so I'm just waiting for Autumn and Julia.
this one? Yes. Thank you.
Yeah, there's a folder which is GSI 2025. Maybe it goes down a little bit. Yeah,
GSI
this,
top?
yeah, this one.
This one?
Yeah.
Do you want to see it?
Yeah.
Let me. Sorry, I don't use PCs. So I'm like
Yeah.
struggling to figure out how that. Yep. I just want to be able to eject it. Okay, we'll worry about that later.
You dragging it into this one?
Yeah.
So I'll have this timer.
Yes.
Yes.
Good.
Mhm, good.
Okay. And then I think he's talking about the timer. Yes.
Okay.
So 8 minutes and then 2 minutes question. I'll introduce your name and title and have you come up with this.
Elena.
Smoothly.
Hi. Ooh, this is a little guy.
I know.
And you guys are all competing as a group for a prize. I'll talk about that. So,
No pressure, Nelson.
No pressure.
I know, right? This one?
Yes, it's that one. Yeah.
Why did that happen? This one, correct?
Yeah.
It's weird. Okay. I'm gonna put it here.
It's good practice, but there's a little.
Oh, you're in Philip's lab.
Yes. Yeah.
I'm like, this looks familiar.
yeah,
Is that supposed to be like that? Is it animated?
it's
Okay,
animated.
okay,
Yeah.
just making sure.
I know it looks crazy. This one also
Same.
looks
Yeah, no, that's
crazy.
okay.
Okay.
So far, so good?
Yep.
Yes?
Looks great.
And I'm pretty sure we can't use presenter notes. So
But that's
if you were
fine.
hoping
That's
for that.
okay. I like have them on there, but I can do it.
Okay, okay.
Thank you.
Excellent. That's what I like to hear. Yeah, let me spit out your thing here. Oh my gosh, this mouse.
Thank you.
Tess, my dear.
Rachel.
How's it going?
I'm excited.
Good.
Just feeling a little silly.
Why?
I don't know.
Oh,
Feeling so adult, you know?
you are an adult.
I guess so.
You look great too.
Aw, thank you.
This one?
Yes, that one.
There's no presenter notes.
Don't.
And you're good at speaking, but some people crutch on them too much and it's bad. Never
Yeah.
use them if you
Yeah.
can avoid it.
Do we have, like, I see here, this is a microphone. Is this a slide advancement?
Yep, this is. Yep.
Sweet.
Okay,
Yay.
wait, I'm like PC illiterate, so this takes me far longer
I am
than
too.
most people.
I mean, it's not like I've never used a flash drive before, but when I had to do it, I was like, am I going to know how to do it?
Right? Do you
Thank
have it
you.
in? Oh, it's in a PowerPoint. Why is yours formatted differently than everybody else's? Let's see what it looks like.
Is it? Like in a bad way?
No, but see how the image looks different than the other ones? It might be
Yeah.
just an older version, I guess. Just tell me if anything looks funky. Are you going to talk about your bread?
No. Oh my gosh. Sometimes I think about that like at night and then feel embarrassed.
Why? It was cute. I thought it was cute.
Aw, well, thank you. Maybe if I present in front of like younger.
Yeah, right. Like the right audience. Yeah.
Yeah.
Okay, it looks good to me. Yes.
Yes.
Okay.
Thank you so much.
And I've ejected you. Okay, good.
Sweet.
Awesome.
Yay.
Hello. Sorry, I'm just organizing them. Hang on a second. I don't know what that is, but that is not ours. Okay. Jillian. Hi, Rachel. Oh, you're number one. Okay. There's no presenter notes.
There's no presenter view. That's okay.
Oh, okay. I thought you were going to freak out for a second.
No,
your
so I get
reaction.
to see it on there. too.
No, it's yes, but because he's doing like a live stream, I guess you can't do both at the same time. I don't
Oh,
know.
that's
Okay.
as
Oh,
long
okay.
as I can see the slides. That's fine.
Yeah. Your reaction though, I was like, oh no, is that going to be bad? Is it this one, GSI 2025?
Yes.
Okay.
Yeah, the rest would just be like tables and nothing fun.
Do you want to? So do you want to see it or no? You good?
Oh yeah, that would be great if we could just get a
Yep.
view. Beautiful. Okay. Yeah, this all looks normal. Yeah.
Make sure nothing. Okay.
Yeah.
Oh, viral trickery. That sounds fun.
Thanks.
All right, let me just pop this out.
And we start at 11, right?
Yes.
Okay.
Wait, let me give you your flash drive.
Oh.
Super. Awesome.
Thank you.
I don't know. I said, like, I told her to like try to go in and resend it. Maybe that would. Oh, this isn't gonna help me.
Just waiting for autumn.
Just autumn.
I'm gonna start the program soon. All right. Good morning, everyone. I'm Nelson Lau, the director of the Genome Science Institute, and I'd like to kick off this 17th annual research symposium of the Genome Science Institute. And I am grateful to all of you coming to this event, which we host every year in November. It's really a celebration of the genetics and genomics research that we do at Boston University. And it's also a great way to bring everyone together to showcase their work. There's a little bit of a competitive element to the symposium. We have a bunch of lightning talks in this morning. And then at noon, we'll have lunch over here to the right with poster sessions that will also be judged for awards. And then after the poster session, please visit all the vendors here providing their genomics and genetics tools that we hope that all of you will partake in. Then our interim dean, Dr. Hyung Park, will give her address before we have new faculty speak at the end of our symposium. We want to thank all the vendors again for supporting our symposium. Their contributions to the symposium pays for all the food and allows us to host this event. In these tough times of funding, we could use all the funding we can get. And I'd also like to give a shout out to 10X Genomics because they are also sponsoring a pilot grant that the applications are due November 16th. And for your application to be really considered, please visit them. Alyssa Atkins over there is hosting a table there to find out more details about this program. It's also run by Yuri Alexeyev, our sequencing core director. Unfortunately, Yuri is not here today, but he's definitely a great resource here for all of our sequencing needs here at BU. Please also keep in mind for the various GSI seminar series that we host. We have 2 seminars in December. Dr. Ruben Dries, who is not here today, will be speaking on December 2nd. And then on December 9th, we have Takara giving a technology seminar. So any students, postdocs, faculty wishing to host a GSI seminar, please reach out to us for the spring 2026 because we have slots open. We definitely would appreciate your assistance in suggesting good people to invite. And then lastly, if you guys came in at the welcome desk Sabina is handing out raffle tickets for you to potentially win one of these nice fleeces from the graduate program in genetics and genomics. We'll do this drawing at the very end of the symposium, so please stay in order to be able to win your prize. If you're not here and we draw your ticket, we'll then move on to the next ticket. So without further ado, let's start off with our training lightning talks. Here's the schedule. It might shift around depending on who's running late, but we'll start off with Jillian Petrosky talking about internal RDRP recognition sites triggering 5' something synthesis in filoviruses. All right. And your seminar, where's your talk? Can you find it? All right.
All right. Can everyone hear me? Okay, great.
Here's the laser pointer.
Oh, awesome. Okay, thank you. The something is 5' copyback DVGs. And today I will be telling you just a small part of the role that they've played in my thesis work so far. I'm currently a 4th year in the bioinformatics program, and we'll get started. So hi, I'm Jill. I am in the Connor lab over at the Needle, which is a very interdisciplinary group of virologists, pharmacologists, and bioinformaticians. The lab has lots of interests, but I'm going to simplify them into viral evolution and viral trickery. I personally love filoviruses. And so filoviruses have single-stranded RNA genomes, and they have several very famous members that I'm sure some of you are familiar with, namely Ebola virus, which is known to cause regular outbreaks with fairly high human case fatality rates. Today, I will be talking about a lesser-known filovirus, Thai forest virus. And so Thai forest emerged in 1994 in the Ivory Coast. It's only known to have caused one human infection, which was survived. Only recently has it begun to be used in animal models in the laboratory. And this is because it's a valuable mixed survival model. So we can compare those that survive and those that succumb, unlike some other phelos. And so how this whole project began was I was tasked with looking for single nucleotide variants in fatal Thai forest virus infections. And to do so, I looked at next generation sequencing data that had been collected from macaques that had been infected with Thai forest virus. And then we sequenced whole blood samples that were taken throughout the course of infection. And then did my normal NGS pipeline and started with a very basic step of aligning all of those viral reads to the viral reference genome to look at the read depth across the entire genome. And aside from those two big dips, which are structured regions of the genome that are notoriously hard to sequence, we had fairly even coverage throughout the entirety of it. Oh, sorry. However, when I looked at terminal samples, I was seeing this kind of unique and unexpected coverage phenotype with a huge increase in coverage at the very end of the genome. And so now my story about viral evolution becomes one about viral trickery. So what could be causing the 5' end to be made more than the rest? Enter defective viral genomes, or DVGs. DVGs are truncated viral genomes that are generated when replication doesn't entirely get finished. There's several varieties, but I'll talk about one today. So normally when you're replicating, the viral polymerase sits down on an antigenomic template strand and it chugs along and it produces a full-length genome. And this is what gives you, for the most part, even coverage across the whole viral reference genome. But in the case of a defective viral genome, the polymerase departs that template strand at what's called the breakpoint, and then it falls onto the nascent strand it was creating at a position called the reinitiation site. And then it continues to process down that nascent strand and create a very short hairpinned RNA known as a copyback defective viral genome. And what this is basically the 5' end of the genome copied over and over again. And so when you have lots of them in your sample, that is why you get that huge increase in coverage at the very end of the genome. And so defective viral genomes in the virology space, until I would say probably like the early 2000s, were a fairly ignored phenomenon and primarily thought of as a thing that only happened in tissue culture when you're working with stocks that had been domesticated over time. And however, current research is showing that, in fact, they are being made in the process of authentic in vivo infections. And they've been linked to viral persistence and immune activation, which may be advantageous for the virus. The exact cause of that polymerase template switching event that creates a copyback DVG remains unknown. And so my early hypothesis was that DVG formation must be a normal part of fatal typhorus virus infections. And so to look at them, I went back to that next generation sequencing data set, which normally we would QC, we would align to the host to get rid of host reads, and then we would finally align to the virus, take those viral reads, and look for the single nucleotide variants. But to look for DVGs, you have to go through that unmatched reads garbage pile. And then I use a tool called DI Tector, which takes those unmatched reads and iteratively segments them and realigns those pieces to the viral reference genome until it gets a best mapped pair. Based off of where those pairs are and the sense of the fragments, we can tell what type of defective viral genome is present and its breakpoint and reinitiation site. And this gives you a unique DVG species. And so when I put my typhorus virus data into DI Tector, I saw that overall coverage increased, the abundance of 5' copyback DVGs increased throughout the course of infection, which was really cool. And then I was curious about where those defective viral genomes came from. Since people think that they're coming from the stock, I thought I would compare the number of species between the stock, all of the pooled day 7 samples, and all of the terminal samples. And we see that there's around 400 unique species to start off with in our stock. So they are happening in cell culture and in stock. However, by the time day 7 rolls around, most of those species have been eliminated and new ones are emerging. And then that continues later on in infection. So we know that 5' copyback DVG synthesis is a spontaneous process that is naturally occurring in typhorus virus infection. So hypothesis 1, we've checked that off the list. But how are these DVGs being formed? And so when I look at the junction sites, that breakpoint and reinitiation site for all of those terminal time point DVGs, I see them all clustering at the 5' end of the genome. And that's not necessarily surprising, but when we zoom in on that position specifically and we plot things a little differently, the way this plot works is the positions where the arcs are crossing the x-axis are your breakpoints and reinitiation sites, and then the height of the arc indicates the number of reads mapping to that DVG species. We see that 78% of the copyback reads in those terminal samples have reinitiation sites within a small 20 base window. What could this be? So hypothesis 2 is that something within that sequence is driving this copybacking phenomenon. And so when you think back to a few minutes ago when I showed you how copybacks are made, that reinitiation site where the polymerase falls down onto is a place where strand synthesis is beginning again. And normally in a virus, strand synthesis begins at those very ends of the genome, at the genomic and antigenomic promoter. And so my thought was that if you compared all 3 of those sequences to each other, there should perhaps be a shared motif across them. And I used meme to do that. And I find this poly-U shared motif across all 3 sites, indicating that the reinitiation site hotspot resembles those promoters. And this leads me to a new proposed model for 5' copyback DVG synthesis in filoviruses, where an internal promoter-like region distracts the polymerase that is actively replicating and creates the copybacking effect. So in summary, I've shown you that DVG formation appears to be a normal part of typhorus virus infections. Typhorus virus has a hotspot of these reinitiation events internal to it. This reinitiation hotspot resembles the genomic and antigenomic promoters. And we propose a new sequence similarity-induced template switching event that is responsible for 5' copyback DVG generation. With that, I'd like to thank my lab and our collaborators, and I will take any questions.
Thank you, Jillian. Questions?
Hello. You clearly showed how surprisingly the stocks of the DGV form in the stock versus the infection and late infection are surprisingly different. But later you show the arts plot that you have a very few set of dominant species.
Yes.
Are those the ones in common within the stocks or the ones that are dominating?
They're dominating in terminal time points.
And they are different.
Yeah, if we go
they
back.
Are not shared at all.
Sorry.
Yeah.
Oh, very different. We only have one shared species across all time points, and it was not a common one.
It's not the
Yeah.
very high one. Okay, thank you.
More question over there?
That was a really great talk, Julian. Thank you. I just wanted to kind of maybe challenge your hypothesis about what's happening in terms of the promoter-like sequence.
Mm-hmm.
And you said that might be interrupting normal synthesis. But when this polymerase is synthesizing, it's synthesizing, if I'm understanding correctly, the opposite strand. So the polymerase would see the promoter in a template strand, but now you're saying it can see the promoter sequence in a transcript strand, in a newly synthesized strand as well?
Correct. I'm not sure. I guess to pose a slightly different hypothesis, I'm not sure if it is directly causal for pulling it off course. But once the polymerase departs for whatever reason, whether it be able to see behind it or something else has caused it to fall off, that site that resembles a start of a genome seems to be attractive for reinitiating synthesis.
That makes sense. Yeah, I like that. Thank you.
Okay, let's thank Jillian and move on to the next speaker, who will be Benedetta D'Elia, speaking about characterizing the effect of endogenous and exogenous stress on viral cis-regulatory elements. So following along the virus theme here.
Hi, my name is Benedetta, and I'm a third-year PhD candidate in the Fuchsman-Bass lab. And today I'm going to be talking about my project where I'm trying to understand how stress may lead to viral reactivation and pathogenesis. So viruses that establish chronic and latent infections are a major public health challenge. And many of them, as we know, are associated to conditions such as cancer, autoimmune, and neurodegenerative diseases. And here you can just see a few examples that are, common, such as the link between cervical cancer and HPV. The viruses have a complex replication cycle in which, upon entering the cell, they can basically integrate into the genome or remain episomal. They can establish latency and later reactivate to then initiate lytic replication. So because of that, viruses basically need to continuously adapt their gene expression programs in order to persist in the host and in order to evade immune defenses, as well as to propagate into other hosts and other cells. For example, in the course of an infection, we know that viruses are exposed to both endogenous and exogenous stress signals, and that may activate transcriptional programs that can result in cell death and pathogenesis. We know that these signals can be intracellular, so they can come from the infection in itself. And for example, they can be mitochondrial stress or endoplasmatic reticulum stress, as well as metabolic perturbations. And there can also be environmental signals, such as drugs or hormones. We already know from other studies that stress can cause viral reactivation. For example, herpes simplex virus 1 can be reactivated by heat shock. Epstein-Barr virus can be reactivated by hypoxia, and human cytomegalovirus by oxidative stress. But despite all this clinical and functional evidence, we still don't really know how a lot of these mechanisms actually work. Because viruses have a compact genome that is a lot more compact than human genomes, they cannot encode for all the protein sensors to detect all possible cell states that they might encounter in the course of an infection. So one way to sort of overcome this limitation is basically the sensing of host transcription factors downstream of cellular signaling pathways through viral cis-regulatory elements that I'm going to refer to as viral Cres from now on. Viral Cres are short stretches of DNA that modulate transcription. And so basically, what viruses can do is that by binding host TFs at these sites, they can dynamically regulate gene expression, and they can also find ways to persist in the host and switch their different stages between latency, reactivation, active replication, and immune evasion. Until recently, less than 200 viral CREs were known, but our lab has identified over 2,000 active elements across over 30 viruses. What we still don't know is which viral CREs can be activated by stress and how does that really work. So in order to investigate that in a comprehensive manner, we have employed massively parallel reporter assays to basically test the impact of stress on 27 double-stranded DNA viruses and 7 retroviruses that infect and cause disease in humans. For this purpose, we have basically decided to test viral genome activity by splitting the genome into 200 base pair tiles that overlap by 150 base pairs. In NP arrays, viral genomic fragments are cloned upstream of a minimal promoter, reporter gene, as well as unique barcodes. And then transcriptional activity of each fragment is inferred by comparing the relative number of barcode reads of the RNA fraction versus the plasmid fraction. So in this way, we can test tens of thousands of sequences simultaneously, and that is what we have done with stress. So we have measured the impact of different types of stresses like chemotherapeutic drugs, apoptotic agents, and so on 34 viruses. So here, I'm basically showing you a differential activity map of human cytomegalovirus that is, whose pathogenesis is basically characterized by immune dysregulation, inflammation, as well as neurodegeneration. So you can see here a bunch of different treatments. Dexamethasone is a glucocorticoid medication. ABT-737 is an apoptotic agent. Doxorubicin, DNA damaging agent. Hydrogen peroxide triggers the oxidative stress response. And G132 is a proteasome inhibitor, oligomycin a mitochondrial inhibitor, and we've also tested heat stress at 42 degrees. On the x-axis, you can see genomic position, and you can see in the bottom track genes, annotated genes of HCMV. And then the top tracks in green are control tiles, whereas red and blue marks represent activated or repressed tiles in response to treatment compared to control. So in general, we can observe that HCMV is rich in stress-responsive viral CREs, and more specific results, some patterns that we see is that MG132 has a broad repressive effect, whereas heat stress has a broad activating effect, and other ligands seem to have more specific responses. We've also tested some specific hits, and we were able to recapitulate some known results. For example, the major immediate early promoter of HCMV is already known to be responding to dexamethasone through a glucocorticoid response element. And we've also been able to see that the other 3 regions in this figure have glucocorticoid receptor binding sites. And all of these genes are immediate early genes that we think may be involved in HCMV reactivation and latency. But the question is, do other viruses also respond to stress signals given what we've seen? And the answer here is yes. So we can see in this heat map that shows differentially responsive viral CREs in log 10 scale that basically different viral families respond to different stresses in different patterns. For example, we can see that herpes viruses and adenoviruses are broadly responsive to a wide range of stresses, and other families like papillomaviruses are not as responsive and are more selective. And finally, we've also seen that stressors can modulate both overlapping and unique sets of viral CREs. So most viral CREs actually seem to be regulated in a unique manner by the stressors, but we also do see that some of the stresses can instead modulate multiple viral CREs. So in summary, viral CREs are broadly stress responsive. some virocrease act as shared stress sensing hubs, and others respond to more specific pathways. And mapping these responses points to mechanisms that control latency, reactivation, and stress-driven gene expression. And I would just like to thank my lab, my advisor, Juan, for the guidance and support, and my committee members. Thank you.
Questions?
Hi, very nice talk. I'm interested about treatment with proteasome inhibitor, which seems to have the largest response. Are there many transcription factors downstream of that, or any specifics that you can say?
So yeah, that's a great question. We are actually currently in the process of doing those follow-ups. We are looking at the transcription factors, motifs, the general regulatory network. In the case of NG132, we do see a really broad enrichment of SP sites in the tiles that are downregulated by this type of stress. We don't exactly know why that is, but we hypothesize that it might just have a really broad repressive effect on transcription, as it has already been, described by other studies. But yeah, we definitely aim to continue looking into that. Hi.
Was there many obvious candidates for following up in the context of the viral infection? Have you looked at several CREs of the virus that implemented infect cells and what the impact of stress in the viral
Yeah. So actually, I'm currently in the process of doing several follow-ups for interesting candidates. One of these is actually HCMV, and we have established a collaboration with someone over at the Lerner Institute in Ohio that is an expert in HCMV infection. So yeah, the plan is basically to basically infect cells with HCMV and our viral library at different time points and try to establish a time course of infection. And we're currently working to optimize, those experiments. But yeah, that's the plan.
Okay, thank you, Benedetta. Move on to the next speaker, Elena Yusun, also from Department of Biology. She'll be speaking about CAR induction of the Wnt pathway receptor, FCZD10, during the early stage of hepatocellular carcinogenesis.
Yeah, good morning, everyone. I'm Elena, and I'm from Dr. Waxman's lab. And today I'm very happy to be here to share a very cool research topic, which is CAR introduction of the Wnt pathway receptor friso-10 during early stage of hepatocellular carcinogenesis. So what is CAR? Actually, CAR is constitutive angel state receptor, and it can bind with TC pop-up, which is a foreign chemical and agonist ligand. And after the binding, the CAR will translocate to the nucleus and then activate certain genes. So this pathway actually can lead to plays multiple roles in liver metabolism and disease. For example, xenobiotic metabolism, lipid and glucose metabolism, and cell proliferation, and of course, tumor genesis. So these slides actually shows that the liver cancer is a step-by-step process. So we'll start from the healthy liver. So we can see that after the TC pop-up bind with the CAR, it will lead to fatty liver in 2 to 4 weeks of the TC pop-up exposure. And then some immune cells will get activated and then lead to fibrosis and cirrhosis, and finally lead to liver cancer after 14 to 20 weeks exposure. However, the metabolism is not well understood. So among all of the genes that are induced by TC pop-up, friso-10 is a really interesting target because we can see right here, it's upregulated above 100-fold change after 1 day and 2 week TC pop-up exposure. And this kind of introduction can persist along the whole process of the tumor genesis. So what is friso-10? Actually, friso-10 is a receptor of the Wnt pathway. So we can see right here, when there's no Wnt signaling, the beta-catenin will get degraded, so no genes are opened up. However, when friso-10, when Wnt bind with the friso-10 under TC pop-up exposure, the beta-catenin signaling pathway will spare the beta-catenin from degradation. So there are many genes that are involved in liver tumor will be transcribed. So our hypothesis is that the CAR activation drives tumor genesis by upregulating frisoten and then activate the Wnt signaling pathway. The approaches we use is that firstly, we identified CAR responsive genes that may increase the activity of the Wnt pathway. And also we use single nuclear RNA-seq data and single nuclear attack-seq data to identify the Wnt pathway-related genes, which is responsive to TC pop-up, and we use histological staining to validate that. So our next question is that how does CAR induce the frisoten? So right here we can see that after a 2-week TC pop-up exposure, there are 5 chromatin sites that open in the TC pop-up exposure liver, and some of them shows a higher DHS as early as 3 hours and 27 hours. And the ChIP-seq data shows that the K27 acylation, which is a marker of the enhancer, also increased at 27 hours. And actually, these sites are CAR binding sites. So we can conclude that the TC pop-up induced the chromatin opening within 3 hours at sites with CAR binding and strong increase in enhancer markers. So here is our hypothesis is that after binding with TC pop-up, the CAR will bind to frisoten enhancers and then seduce the frisoten expression. And then the whole Wnt pathway will get activated. So there are some genes that are involved tumor get transcribed. So here is a single nuclear RNA-seq UMAP. We can see that the frisoten is induced by TC pop-up in a small number of pericentral hepatocytes. So we can see that these red dots are frisoten positive cells, and the blue dots are GLUL, which is a target gene of the pericentral hepatocytes. So in this case, we only focused on the pericentral hepatocytes. So we reclustered them and we get 12 subclusters. And then we compared the frisoten positive cluster versus the negative. And we get 18 genes that are enriched in Wnt pathway. And some of them will activate the Wnt pathway and some of them will inhibit. But when we look at the GLUL, which is a target gene of the Wnt pathway, we can see that the GLUL has a much higher expression in the frisoten positive cells versus the negative cells. And among all of these 18 genes, SOX4 is a really interesting target because it is a cancer stem cell markers and it directly interacts with beta-catenin and have some binding sites with the TCF. So right here, we can see that similar to frisoten, SOX4 is induced by TC pop-up from 2-week to 22-week, but not from 1 day. And also, SOX4 is mainly expressed in the pericentral hepatocytes with a higher chromatin accessibility in the TC pop-up pericentral hepatocytes. Another thing we need to mention is that within frisoten positive cells, we can see that the SOX4 chromatin accessibility is invalidated, which means SOX4 and frisoten may co-express. So considering that, our next step is that we did a differential gene expression between the cells that either expressed SOX4 positive or frisoten positive versus double negative. And we get 233 genes. And the David enrichment analysis shows that these genes are enriched in cell cycle and DNA replication pathways. And IPA result shows that the ELDR and TBX3 are promising upstream regulators whose function is maintaining the stemness of the cancer stem cells. So here is our IHC staining result. We can see that it's very clear in the TC pop-up tumor samples. These brown dots are frisoten, but it's not very clear in the TC pop-up liver, although we observed some like brown dots. So our next step is that we will use SM fish to validate that. And another very exciting news is that we carried out a single nuclear RNA-seq using PIPE. And we carried out 4 samples, which is 22-week control liver, 14 and 22-week TC pop-up liver, and 22-week tumor. And thanks to GSI grant, so we can evaluate this new technology. And we expect it to get results in a few weeks. So our conclusion is that TC pop-up activate of CAR strongly induced the frisoten in a minor population of pericentral hepatocytes, which we propose acquires cancer stem cell-like properties, at least because of the Wnt beta-catenin signaling pathway upregulation, and activates a certain population of the targets, which are related to cell cycle and DNA replication. So frisoten is a promising target for early stage liver cancer prevention or treatment. So finally, I would like to thank everybody in Dr. Waxman's lab, especially my tutor, Dr. Waxman's support. And we're also very appreciate the help from BU Single Cell Sequencing Core, GSI, and Illumina. So do you have any questions?
It's fantastic. Right on time, too. You had extra time. Any questions from the audience? If not, I'll start off with one. Oh, okay, Darren.
Interesting. Okay, I'm a metabolism person, so my brain stood up when you mentioned the I think glucose 6-phosphatase at the beginning, and I was just wondering if that is down-regulated so that such cells would, rather than exporting glucose, would use it for their own, synthesis of cell constituents, et cetera.
Sorry, I cannot hear you.
I think an early slide said there was something about glucose 6-phosphatase, in other words, an effect on carbohydrate and energy metabolism. And I was wondering if the effect, in contrast to a number of these other genes that are upregulated, if that was downregulated such that such cells would no longer export glucose, but rather would have enhanced metabolism for proliferation.
Yes, that's a very good question. And we don't know the answer about the glucose, but for the weight coupling pathways, we do observe that some genes are related to cell cycle. But yeah, I'm not very sure about the glucose related.
Okay, thank you.
Okay, we're gonna shift gears a little bit to, although Autumn is next, we haven't got our presentation loaded up yet. So we're gonna move next to Emily Kirshner. Autumn will go at the end, and then we will continue on with these lightning talks. Emily will be talking about metabolic control of histone methylation in learning and Alzheimer's disease.
Okay. Hi, everyone. So I'm in the MEVS lab, and in the MEVS lab, we work in the brain. So when we think about how a memory is encoded upon a learning stimulus, you get activation of a subset of neurons, and through repeated stimulation and recall events, you end up strengthening that ensemble and eventually resulting in an encoded memory. So activated neurons undergo dynamic changes in chromatin structure, which allows for the rapid induction of immediate early genes. Immediate early genes are rapidly transcribed with neuronal stimulation, and then they go on to regulate secondary response gene programs as well as modulate synaptic plasticity. This whole process is facilitated through epigenetic mechanisms. So histone methylation is a very nuanced mark. It can either be activating or repressive. And it has already been shown that histone methylation is actually critical for memory encoding. So here I'm just showing an increase in H3K4 trimethyl after learning in the hippocampus of mice, as well as if you deplete that mark with MLO1 loss, which is a methyltransferase, you get reduction in memory performance as shown here in T-maze and with the orange bar. So histone methylation, it can either occur on arginine or lysine residues on histone tails. So here again, I'm showing that activating mark K4 trimethyl, and certain subclasses of methyltransferases put on these modifications. And actually histone methylation is intricately linked to metabolism as S-adenosylmethionine or SAM serves as the universal methyl donor. SAM is synthesized by MAT2A or methionine adenosyl transferase 2A. And while the connection with SAM and epigenetics is pretty well established, the actual molecular mechanisms of how this occurs, especially in the brain, is pretty poorly understood. So we have found that MAT2A is expressed in the nucleus of differentiated N2A neurons, which kind of brings us to a key mechanistic question, which is, does MAT2A localize to chromatin to provide SAM for histone methylation? So to answer if MAT2A binds to chromatin, again, we're using that differentiated neuroblastoma cell model. And we performed cut and run against MAT2A. And we actually did find that, yes, MAT2A does bind to chromatin at activity-induced genes, so ARC and FOS in the promoter regions. And in fact, this binding also overlaps with known K4 trimethyl regulated loci. So that was really exciting. We also wanted to ask if MAT2A interacts with any chromatin modifying enzymes. So we performed this technique called RHYME, which works a little bit similar to CHIP. You use antibody conjugated beads against your protein of interest, in this case MAT2A. And instead of a DNA readout, you actually cross-link all your proteins together and then do mass spec on that eluent. So this worked great. Our top hit was MAT2A as expected. Second hit was MAT2B, which is the regulatory subunit. And we actually got a whole slew of methyltransferases as well, so we can confirm yes, MAT2A is interacting with these chromatin modifying enzymes. So I wanted to see if this MAT2A binding also held true in vivo. So now we moved into the mouse, and I performed CHIP against MAT2A and the mouse nucleus accumbens. And we saw the same thing. MAT2A is binding to chromatin at gene regulatory elements, mostly promoter driven. And in fact, this binding is enriched for themes like genome maintenance and chromatin architecture. So we're still parsing through this data a little bit. We also wanted to know if there would be any sort of behavioral readout with MAT2A regulation. So I performed stereotactic surgery to knock down MAT2A in the mouse dorsal hippocampus, and then performed a battery of memory assays. So we did get good, efficient knockdown, as you can see here with this Western blot image and the quantification of that, as well as expression of our GFP reporter. We also saw a trend in reduction of H3K4 dimethylation. So we see methylation deficits with this knockdown as well. When we looked at the memory readouts, so we tested for recognition memory using novel object recognition, and we did see a really significant deficit in our knockdown animals compared to controls. We also put them through contextual fear conditioning, where they get a foot shock in a certain context. You put them back in the box and kind of test their freezing behavior. If they remember they got a foot shock there, they will freeze more. And we did see a deficit in our knockdown animals as well. We also tested them for location memory, which is a little bit more of a spatial paradigm. And we see a trend in object location memory there too. So from this data, we can conclude that MAT2A knockdown results in memory deficits. So 1-carbon metabolism is a really complicated cycle. It's essential for cell survival, but it actually already has been implicated in disorders of learning and memory. The biggest one, Alzheimer's disease, which I'm sure we're all familiar with. So SAM, that metabolite, is actually down in AD human brains, kind of across the board, every region. And AD model and human brains also exhibit this DNA hypomethylation effect that actually, if you supplement with SAM, that metabolite, you can rescue some of those deficits. However, how this process regulates histone methylation is pretty underexplored, so that's where my project kind of ties in. So to answer that, we're using the APP-SA knock-in mouse model. This mouse has a humanized APP sequence as well as 3 familial AD mutations. They exhibit robust amyloid beta pathology as well as the metabolic deficits that we see in Alzheimer's patients. So this mouse is pretty new, so I just wanted to characterize it at 6 months of age, just to start off the bat. And we did see cognitive deficits at this age in contextual fear conditioning, which was awesome because we have a phenotype to go off of. I also collected hippocampal tissue and ran a Western blot for some histone modifications, and we do see a trend in reduced H3K9 methylation, which is a canonical repressive mark. And then finally, because we know that SAM supplementation has therapeutic potential, I wanted to test if MAT2A upregulation could also alter memory. So what I did was, again, perform stereotax to overexpress MAT2A in neuronal nuclei in the dorsal hippocampus at 4 months of age, so before severe amyloid pathology, and then tested them at 6 months of age. So we got great overexpression in these animals. You can see the endogenous MAT2A band here, as well as that strong overexpression there. It's just a little bit heavier because of our GFP and B5 tags. And then actually, we also see an enhancement of freezing behavior in these mice when compared to the APP-SA non-surgerized controls. So we're actively adding animals to this analysis, but I'm very hopeful. So to conclude, MAT2A is binding to chromatin in the brain at plasticity-related genes. Downregulation of this process results in memory deficits, and then actually if you upregulate this process in the hippocampus of this APP cell Alzheimer's mouse model, you can enhance memory. So for some future directions, we're currently in the process of analyzing some MAT2A chip data from this mouse model to map MAT2A genome binding. I have plans to use spatial transcriptomics to test how MAT2A regulates gene programs in direct spatial proximity to amyloid aggregates. And I also want to know if SAM supplementation can rescue molecular deficits, mostly those gene expression readouts. So with that, I'd just like to thank the whole MEVS lab, as well as my department, PPB, GSI, GPGG, our funding sources. And then if you want to learn more about this project, I'll actually be giving the GSI student seminar on Tuesday of next week. So I'll be giving an expanded talk, as well as if you want to learn more about the lab, my lab mate Natalie is presenting a poster. So I invite you to visit her then. Thank you, and I'll take any questions.
Hi, that was very interesting. Can you distinguish, your model showed those little SAMs right in the vicinity. So do you actually think that the localization to those chromatin sites is to provide SAM locally for the methyltransferases? So for example, can you express an enzymatically defective version of MAT2A and does it still localize? Or is it possible that MAT2A has additional chromatin modifying functions or transcription or epigenetic regulatory functions per se?
That's an excellent question. So that is a part of our hypothesis that this MAT2A dependent histone methylation does depend on that like localization to chromatin to provide SAM right there to be tacked onto histones. We do actually have a vector with a catalytically dead MAT2A as well, so we're using that. I think that is our hypothesis right now, but SAM is used for a whole slew of things naturally. So we know that DNA gets methylated, RNA gets methylated, so we're not open. We're open to exploring those further as well, but right now focused on the histone side.
Emily, you mentioned actually deficits. You mentioned MLL mouse that had a phenotype, right? And MLL is connected to H3K4 methylation. And then you had some data on H3K4 methylation, but then you showed in the mouse, you showed H3K9, and also you showed interacting partners, right? Was because nomenclature for set for H3K4 methyltransferases, it could be different. Are those interacting partners MLL that was shown deficit in the publication before or not?
Sorry, are you asking if our like RIME outputs that interact with MAT2A are also interacting with MLL, or did we get MLL1 on that
Did you
list?
get MLL? Because MLL was functionally implicated, right?
Right.
So I'm wondering if you mechanistically connect.
Okay. I don't think we got MLL1. However, we did get other K4 methylation enzymes. We actually got a lot of enzymes that all put on different modifications. So it's definitely true. Like I said, SAM kind of contributes to everything. But we do have this theory about basal turnover rates and histone modifications and how some sites may be more sensitive to MAT2A activity levels than others.
Thank you.
Okay, thank you, Emily. I'll move on now to Teresa Rosado, who'll be speaking about investigating the mechanism of PAX3, post-transcriptional gene regulation, driving melanoma progression.
Thank you. Hello, everyone. PAX3 is critical to melanocyte development as well as melanoma progression. We know that PAX3 is upregulated in over 75% of all melanomas, and it has also been shown that loss of function of PAX3 is detrimental to melanoma, both in vivo and in melanoma cell lines. Now, PAX3 is best known for its role as a transcription factor, and you'll see here it consists of two major transcriptional binding domains, the paired domain and the homeodomain. Interestingly, the Lang lab has found a non-canonical function of PAX3 that is completely independent from its function as a transcription factor. We now know that PAX3 performs post-transcriptional gene regulation through its mediation of the nonsense-mediated decay pathway, or NMD for short. So NMD is actually an mRNA quality control pathway that targets transcripts that contain premature stop codons that are proximal to exon junction complexes, or EJCs. Now, EJCs are necessary for most NMD pathways, and they consist of a couple core proteins, including MAGO, EIF4A2, and RBM8A. And the Lang lab has also shown that PAX3 interacts with these EJC proteins. So this establishes the hypothesis that PAX3 is interacting with EJC proteins in order to increase NMD activity and contribute to melanoma progression. So I wanted to first identify which region of PAX3 is responsible for eliciting NMD activity. And then I also wanted to determine if this is what's responsible for EJC binding. So just to briefly go over the methodology I mainly employed, we use luciferase assays, and how our luciferase reporter works, it's actually the inverse of what you would expect readout-wise, where you'll see here that smaller bars on the bar graphs indicate higher NMD activity. And you'll notice over here on the right, we have our mock in the leftmost column, and then our canonical full-length PAX3 in the rightmost. And again, you'll notice that bar is quite small. So we began by creating a number of large deletion mutants of PAX3. Once more, you'll see the parent domain and the homeodomain, and then the region that lies in between them, the linker region. And what we noticed is that loss of the linker region led to loss of NMD activity. So to zoom in on that region further, here you'll see the amino acid sequence. In blue, you'll see the area, the end of the paired domain. In orange, the beginning of the homeodomain. And then all the other colors here indicate sites of interest to us in trying to identify the NMD domain. So in yellow, you'll see the octopeptide region. This is highly conserved and known for protein-protein interactions. Each of these blue dots is a serine that we know to be phosphorylation targeted. And finally, we'll see in pink the polar disordered region, or that's what we're calling it. It was first identified as a region of low complexity through SEG-like detection software. Simultaneously, we have this theoretical AlphaFold model here that suggests it may be a weak helix. So while we don't yet understand the structure of this region well, it was definitely still of interest to us in trying to identify the NMD domain. Now, we employed the same strategy here where we made mutations to these regions of interest and then measured levels of NMD activity. Starting with the octopeptide region, we saw no change in NMD. Similarly, when we looked at those phospho-targeted serines, we saw no change. However, when we mutated the polar disordered region, we did notice a change. We started once more with point mutations, and we were inserting prolines in hopes that this would mess with the theoretical helix structure there. Again, that didn't impact NMD activity. But when we made a large deletion to this region, we did see loss of NMD activity comparable to that of our mock. And we call this mutant delta H. So before we could feel confident saying that this region was in fact the NMD domain, we wanted to make sure that it did not impact the canonical function of PAX3 in its role as a transcription factor. So to do that, we performed a different kind of luciferase assay with an MITF reporter. This is a known transcriptional target of PAX3. Additionally, here you'll see an extra mutant. This is PAX3WBN8, which we know to be transcriptionally inept. So in this luciferase assay, notably, you'll notice canonical PAX3 is comparable in its transcriptional activity to our delta H mutant. And furthermore, when we look at WBN8 and canonical PAX3 and NMD activity, it's high, but for our delta helix mutant, we don't see that. So with this information, we feel confident that the NMD domain is located within the polar disordered region of the linker region. And it was from this I hypothesized that loss of this region will lead to loss of EJC binding. But we actually found the opposite to be true. I performed a proximity ligation assay where the readout of this is puncta, all of those red dots you'll see here. And each one of these represent either a protein-protein interaction or two proteins very close to one another. And so up top in the rightmost column, you'll see PAX3 and MAGO, again, one of our EJC core proteins. And in our canonical PAX3, you'll see puncta, but also it's quantified to the right. Interestingly, just below it, here we have our delta H mutant. And not only do we see puncta, but we also see that in higher levels than our canonical PAX3. So this leads us to believe that the NMD binding domain is necessary for eliciting NMD, but it's not necessary for EJC binding. So what I'd like to do going forward is identify the NMD domain binding partners that are necessary for activating NMD, as well as locate the region within PAX3 that's responsible for binding EJC proteins. And of course, we do this to better understand the PAX3 NMD mechanism, such that we can later create druggable targets and disrupt melanoma progression. Oops. So I'd just like to take a moment to thank my lab for their ongoing support, all of our collaborators, as well as the American Cancer Society for supporting me both in the lab and through their ACS DICER program. Thank you so much for your attention.
Any questions for Teresa? I'll start off with one. Have you looked at the interaction with PAX3 and MAGO in a melanoma cell line rather than just the HEC cells?
Yes, so actually another member of my lab has done extensive work with PAX3 in melanoma cell lines, and we've actually verified their interaction through 3 different methodologies, so we feel pretty confident there.
Okay. Great. Okay. If there are no other questions, we're going to go on to the last speaker, Autumn Grimmins from the Lions Lab, and she'll be talking about short 5' UTRs that allow for EIF4E independent translation of negative sense RNA viruses.
All set? All right, thank you so much. I'm just going to jump right into this. So, better now?
Speak into that.
All right, this is okay. Okay, so when we think about antivirals, there's generally 2 approaches to designing them. The most common approach for this is virus targeting antivirals. These will target a viral protein that is necessary for replication. However, because of that